Okay, so what is the purpose of conducting analytical method validation? Because whatever method is under evaluation must meet the intended purpose. And that is what the objective of developing and validating the testing procedure. But how one can confirm that those end objectives are met? And that's where the performance characteristics needs to be understood and should be part of your protocol. Hi, my name is Bhaskar Napte. I am the founder of Pharma Growth Hub, and I am on the mission to provide absolute clarity on such a very important technical topic to the pharmaceutical professionals. So, if you are working in the pharmaceutical industry, maybe QA, QC, manufacturing, RA, or analytical R&D, you are most welcome to join this platform. You can find the details about this platform in the description below. So, please check out and decide whether this platform is suitable for you as well. So, as a part of this video, we'll try to understand the performance characteristics for analytical method validation. So, what is the validation means? So, a validation study is designed to provide sufficient evidence that the analytical procedure meets its objective. So, we are going to create some sufficient evidence. And to create those sufficient evidence, we need to understand what are the performance characteristics we need to perform. And this sufficient evidence is going to help us to prove that yes, our analytical procedure meets its objectives. And the objectives could be analyzing related substances or assay or dissolution. Now, these objectives are described with a suitable set of performance characteristics and related performance criteria. This objective could be specificity is the one objective. So the specificity becomes the performance characteristics. And how one can confirm whether the method is specific or not by giving appropriate acceptance criteria to that specific performance characteristics. That when we can call method is a specific, when we can call that there is no interference, something like that. Now, which can vary depending on the intended purpose of the analytical procedure and the specific technology selected. So the acceptance criteria cannot be fixed for all the technologies. It could be different for HPLC than from the UV spectroscopy, or there could be some different acceptance criteria for atomic absorption spectroscopy versus tritimetry. So those specific acceptance criteria will be dependent onto the analytical procedures and the technology selected. Let us now understand the important topic for the today's video, that is the performance characteristics for different test procedures. So these are the four or five different categories of the test procedure. The first one is identity, right? The first one is identity. Uh, okay, so I think let me take this one probably. This will help. The first one is identity is the type of test procedure. The second could be impurity or the purity. And the third could be assay, content or potency. So let us now understand what are the different performance characteristics given according to the ICH guideline Q2R2. That is the purpose of making this video. Okay, so I think let me check now. Okay, so let me zoom out a little so that you will be able to see the table clearly now. And let me take back to the presentation mode. Or right, I think okay. So I hope you now can able to see the complete table. Earlier it was not the full table, so I made a little zooming out so that the table can be seen clearly, completely. So we talked about the three important types of the testing procedure. The first one is identity, the second one is impurity, and the third one is assay or the content potency. Now, what is the important performance characteristics according to the ICH Q2R2? The first one is specificity, right? The first one is specificity. The second one is the range. Third one is accuracy. The fourth one is the precision. And again, the precision can have the repeatability test, and the next one could be intermediate test or the precision test. Okay, so we'll talk about those individual tests as well. But let us now understand, how one can identify that which performance characteristic must be selected according to the type of my test procedure. So whatever plus signs you can see over here now, that indicates that yes, now this parameter needs to be performed. To look at here now for identity test or identification test, like identification by FTIR, UV spectroscopy, or identification by any color development, you need to at least perform which test? Specificity test. Plus sign indicates that this parameter need to be performed. There is no need to perform range as far as identity is concerned. There is no need to perform accuracy, and also there is no need to perform the precision as well. So according to the ICH Q2R2, you need to only perform specificity test for identification test. Rest all test performance characteristics are not required. I think I have another videos on detailed discussion why only specificity is proposed by ICH for identification test. You can always check my videos and try to understand about this one specific very interesting question. The second type of test procedure could be impurity test, like related substances. It could be about other quantitative measurements like residual solvent. It could be elemental impurities. So those falls under the impurity analysis part. So what are the important performance characteristics needs to be studied as a part of the impurity test? Let us now understand the further subcategorization of impurity test, and that is the first one is quantitative test, right? And the second one is the limit test. So what happens in the quantitative test? You actually quantify the content of impurity given in the sample. Report the number that yes, the impurity A in paracetamol tablet is found to be 0.25%. That is called as the quantitative test. Means you are going to quantify that yes, what is the content of impurity A in a given sample. But in case if you are not quantifying the impurity present in a paracetamol tablet, and you only confirm whether the impurity is below certain limit or not. Let us see how the limit of 0.1%. And by the testing procedure, you cannot quantify, but you can only confirm whether it is below your defined specification, that is 0.1%. Example, TLC. You can just compare the size of the or the intensity of the spot, maybe 0.1% impurity A TLC plate versus the paracetamol sample TLC plate, and you'll be comparing the spot at the certain retention value, right? The retardation factor or retention value. And if the intensity of the spot is lower as compared to the intensity of the spot of the reference TLC plate, maybe impurity A, then you can say now this content is less than 0.1% because the intensity of the reference TLC plate comes from the 0.1% impurity A concentration. And as the intensity of the similar spot into the sample TLC plate is less than the reference spot, that indicate that the concentration is less than 0.1%. So that is called as the limit test. I hope you understand the difference between quantitative test and the limit test. So what parameters needs to be performed if we talk about a quantitative test like HPLC analysis for impurity A? The first one is specificity needs to be performed. The second one is the range, that also needs to be performed. Plus sign indicates needs to be performed. You also may have to perform the quantitation limit. Quantitation limit means what? What is the lowest concentration that can be measured with the adequate precision and accuracy. That is the quantitation limit. So especially in case of impurities, you have to also define the quantitation limit, isn't it? What is the another parameter? That is the accuracy. And accuracy can be performed at three different concentrations in triplicate. And it could be from reporting threshold or the quantification level until maybe 120% of the impurity specification. So yes, accuracy also needs to be performed. Precision also needs to be performed. So the precision also needs to be performed, and there could be two different types of precision. One is repeatability, and another could be the intermediate precision. So this both precision studies needs to be performed during the impurity analysis for quantitative test. Impurity analysis for the quantitative test. Let us now you are performing the limit test. You do not quantify the content of impurity in the given sample, but you only confirm whether the content of impurity is below the acceptable limit. That is the limit test. So what parameters needs to be performed? The first one is yes, specificity is required. You need to perform the specificity. The second parameter is the detection limit, right? The second parameter is the detection limit. There is no point in talking about the quantitation limit, because in this particular test we are only confirming whether the sample is meeting the spec or not without having any measurable value. It's only the intensity of the spot, isn't it? And there is no content quantified at the end of the analysis like 0.01% or 0.02%. So if there is no quantification, there is no point talking about a quantitation limit. And the same is the case that accuracy is also not possible for limit test. So accuracy is not performed, and precision is also not performed. Like repeatability and intermediate precision is not possible and performed. So what parameters are performed during the limit test? The first one is specificity, and the second one is the detection limit. Now the similar parameters can be used for the another impurity analysis like residual solvent, elemental impurities, and even other quantitative measurements. And you'll find in the meaning of this one that which follows the similar characteristics as like impurity measurements. Now, what are those test procedures? They are not exactly the related substances or impurities, but they can follow the simple certain same characteristics of the impurities. What is the characteristics of the impurities? Now they are generally available in the sample at very low level, maybe at detection limit or maybe at quantitation limit or maybe below that. That is what expectation or expected outcome during your manufacturing. Let us understand the another activities which also expect the as low as possible content in the sample, and that could be attributed to swab analysis or the residue analysis or the cleaning validation samples. So those samples are always expected to have the as low as content possible. And so for this kind of testing procedures, we can also follow the performance characteristics as mentioned under the impurity test parameter, right? I hope you understand how this can be correlated, and based on to that you can take a decision. The next important type of the test is assay, content or potency. So what parameters need to be performed? And here is the first one. You need to perform the specificity, and it is plus given over here. You need to also perform the range, but there is no need to perform the lower range limit like detection limit or quantitation limit is not required. Accuracy, yes, absolutely is possible and should be performed during the assay. Precision should be performed at both the levels, repeatability as well as the intermediate precision. Now, wherever there is a quantification possible, there is a always accuracy and precision possible and should be performed. So I hope you got a overview on different performance characteristics needs to be performed while validating the different types of the testing procedure. We'll try to understand the meaning of these different annotations now, like one overshare, one again overshare, right? There is a two, and there is a 3 over here. Let us understand what is the meaning of those different annotations. Okay, I think I have reached to end page, but don't worry. So this is the first point. This minus sign indicates what? Minus sign signifies that this test is not normally conducted, right? This test is not normally conducted. So you need not to conduct this test procedure. It is not mandatory or something which is recommended by the ICH. But having said that, there are certain parameters which you may have to think of. Let me take you to the limit test now, or let me take you to the identification test, identity. Now, which parameters needs to be performed? According to the ICH Q2R2, only specificity needs to be performed, isn't it? Only specificity. No response range, no accuracy, no precision. Which is not also possible. Range is not possible, accuracy is not possible. But I can recommend at least to perform the precision parameter also for the identification test. You need not to have the measurement values at the end of the precision study to calculate the percent RSD, but at least can you put the acceptance criteria as yes, if I am performing the precision for identification test for a substance X by FTIR, there may be some acceptance criteria given into the specification. So that acceptance criteria can be applied to the replicate measurements. That if I measure the 6 samples as a part of repeatability, at least I must get all the results meeting to the spec. That is what the expectation. And when someone measures the intermediate precision on another day by another instrument, then again, that analyst also should get all the results meeting the specification. So that is what precision probably could be thought of. What you say, let me know in the comment box. If not range, if not accuracy. Let us move on to the second point, that is the plus sign. So what plus sign indicates? That yes, this parameter needs to be normally conducted, right? Needs to be normally conducted. It's very simple to understand. Now, this mark is what in some complex cases detection limit may be evaluated. Some complex cases detection limit may be evaluated. For example, in case of uh, quantities, in case of what we call mm, impurity analysis, isn't it? In that case, the quantitation limit is important, and hence it is highlighted over here. You can see just on the top or head of the QL there is that particular mark, because for this particular test quantitation limit is also important and shall be evaluated. Oh, in some complex cases DL may also be evaluated. I'm so sorry. So in addition to the quantitation limit, you have to also evaluate the detection limit. That is the meaning of this sign, right? That is the meaning of this sign. It is not about only quantitation limit. It signifies that in case if this sign is available, along with the quantitation limit, you also need to perform detection limit, because that will help you to report the impurities suitably during your analysis. That whether the impurities are below detection limit or not, because that gives a very important information during the analysis. So perform the detection limit as well. QL stand for quantitation limit, isn't it? And DL stand for the detection limit. So it's just a full form. Now this is very important. Annotation 1, number 1 talks about other quantitative measurements can follow the scheme for impurity if the range limit is close to the detection limit or quantitation limit. If the range limit is close to the detection limit or quantitation limit. Now here is the point. This is the number 1, right? So other parameters like I talked about residual, sorry, not what, residue analysis or the swab sample analysis. Now in that case, your lower range limit, look at here, your lower range limit is going to be what now? You will always conduct a range study from maybe quantitation limit until your 150 or 200% of your actual residual limit. In that case, you need to follow this impurity or the purity performance characteristic guidance provided here. So residue analysis is not the impurity analysis, isn't it? It is the cleaning verification or the cleaning validation. Even though it is not exact impurity or purity analysis you're conducting, but you are having the range where your lower range is your quantitation limit. Your lower range is what? The quantitation limit. And in that situations, you have to follow the exact performance characteristics proposed for the impurity or the purity test. This is the point number one. The point number two talks about, and here it is. Okay, so let me again continue further that other quantitative measurements can follow the scheme for impurity if the range limit is close to the detection and quantitation limit. Other quantitative measures can follow the scheme for assay if the range limit is not close to the detection and quantitation limit. And here is the same one letter overshare, isn't it? But now what inference you can draw from here? Because now the lower range limit is not close to either quantitation limit or detection limit. Let us say, let us talk about the another content, not exactly assay content or the potency, but your lower range limit is not needed to be near to the quantitation limit. Means you are not expecting that this particular substance should be available as low as possible. It has to be available at this certain defined content. Maybe, for example, preservative content or maybe antimicrobial content. So those content should not be available as low as possible in the product because they are actually effectively avoiding the oxidization of your substance, and that's where they are called as the antioxidant. And you have the specification maybe for antioxidant like BHA, BST. I don't have exact values in my hand, but I'm just giving the arbitrary values like okay, maybe 100 ppm, right? 100 ppm. Now, in this case, you have defined the value for the BHA content, that is, should be 100 ppm. It is not as low as possible. So, as this is not exactly the assay or content or potency of the drug substance, but this is the substance which acts as a antioxidant. And for such a substance, we need to also follow the performance characteristics as defined under the assay content or potency. This is the meaning of the point number one. Right. So what is the next point? That is the point number two. And what this says? The point number two says some performance characteristics can be substituted with technology inherent specification in the case of certain analytical procedures for physico-chemical properties. Some performance characteristics can be substituted with technology inherent justification. Means technology itself is sufficient enough to conclude the outcome, like mass spectrometry in case of the specific test procedure, or NMR in case of identifying the structure of the compound. So the technology alone can help me to conclude that yes, if I'm using the MS and if I know the mass number is coming so and so, so this is going to be a paracetamol molecule or this is going to be a mesalamine molecule. That is the so some performance characteristics can be substituted with technology inherent justification in the case of certain analytical procedures for physico-chemical properties. Let us move on to the point number two. And this talks about analytical procedure performance characteristics to be demonstrated. I mean, I can certainly demonstrate the specificity of the analytical procedures where mass spectrometry is used just by identifying the mass number. That is, look at here, this is the mass number I got. So this going to be the given substance in the place. So the technology itself is going to help me to justify the specificity. The third point is the lack of specificity of one analytical procedure should be compensated by one or more other supporting analytical procedures unless appropriately justified. Where is the point number three? That is in case of the specificity. So if my current testing procedure is not specific, I'm not able to define this interference. Let us say I'm using the titration for glycoside assay. This is simple acid-based titration. And in this test procedure, I cannot understand whether the method is going to have the specific requires the whether the method is going to acquire the requirement mentioned under the specificity parameters. That means there is no interference coming from maybe the impurities present into the API glycoside. So it is not possible to define the specificity because whatever titrant is going to get consumed may get consumed by impurities also. That is, I am not sure. So I can use the another suitable technique, one more other supporting technique, analytical procedures, where let us say where the related substances are measured accurately. So assay by maybe potentiometry, result is 99%. And if I say that arrest by HPLC, result for the same batch is 1%. I can confirm that. Yes, now look at here. If impurity arrest is actually a specific test procedure, I mean there is no doubt about this 1% related substances result, isn't it? So if I am able to get what is the expected assay result in? If 1% is the RS impurities, the expected results for assay should be 99%. And if my potentiometry gives the same 99% result, that indicates that my potency test procedure is specific in quantifying the glycoside content without any interference from the sample matrix, diluent or degradant or impurities. This is the purpose of the point number 3. The point number 4 is also very useful and important. And what this says, alternatively, a combined approach can be used to evaluate accuracy and precision. So accuracy and precision can be evaluated as a single experiment. You need not to perform precision as a separate experiment and accuracy as a separate experiment. And that has been part of the guideline as well. Suppose if you look into the requirement of precision, it also talks about it can be proved at the 3 different levels. Triplicate preparations, right? Triplicate preparations at 3 concentrations. So this is going to also help you to prove the precision. This is the one approach given in the ICH guideline, that precision can be proved at 3 different concentrations and in the triplicate. So at the end you will have the 9 determinations. What is the accuracy requirement? Accuracy also same, right? Triplicate preparations and 3 concentrations, total 9 determination. So rather than performing accuracy separately and precision separately, you can also combine this study. For example, you are performing the precision and accuracy for assay by HPLC. So you can certainly take a placebo and in the placebo spike 50% of your drug substance and measure the precision. And the same sample can be used for evaluating the accuracy also. The measurement parameters can be different. For precision, you may be using RST, and maybe for accuracy, you may be using percent recovery. So use the same samples for evaluating percent RSD and use the same sample for evaluating percent recovery. So you can save time if you if you combine the precision and accuracy. It is very much accepted according to the ICH as well. The next point is the fifth one. Now, where reproducibility has been performed and intermediate precision can be derived from the reproducibility dataset, an independent study for intermediate precision is not required. Right? Independent study for intermediate precision is not required. Now, how the precision can be demonstrated, by the way? See, precision can be demonstrated at 2 levels. The level 1 is repeatability, right? And level 2 could be either intermediate precision or reproducibility, right? Reproducibility. Now, how you are going to perform the repeatability? It's very simple. You can prepare the six samples by one analyst, understanding the percent RSD and confirming the precision. The second study is intermediate precision. Now, this particular study is performed in the same lab, but by the same second analyst, by the another analyst. If I am doing the repeatability study for six measurement, then I cannot become the part of the intermediate precision. Some another analyst like Vikas is going to perform the intermediate precision on same day or different day, on the different day, by same instrument or different instrument, by using this different set of instruments like HPLC, pH meter, etc. That is called as the intermediate precision. Vikas and I work in the same lab, and that is the intermediate precision. What is the reproducibility? Suppose we have organization with six different manufacturing units and quality control units. I work at Hyderabad location, Vikas work at Bangalore. So if I perform the repeatability by six measurements, and if the Vikas also perform the repeatability, but in the another lab, now in the Bangalore's location, that actually becomes the reproducibility, provided the sample used for conducting the activity is one and the same. So whatever paracetamol tablet batch I am going to use, the same batch must be used by Vikas also at the Bangalore quality control lab. So this is become the reproducibility. So what is important? Either repeatability is must, isn't it? But either you can perform intermediate precision or you can perform the reproducibility. If you perform intermediate precision, then reproducibility is not required. If you perform reproducibility, then again intermediate precision is not required. This is the meaning of the last point, the point number 5. So I hope you understand the performance characteristics of different types of the test procedures, and especially how to identify these annotations, the meaning of different signs provided into this particular table. In case if you have any question about this particular video, please don't hesitate to put into the comment below. Thank you so much.